Good afternoon. Thank you for joining us for our latest Department of Defense Information Analysis Center webinar that's going to cover biological threats. My name is Steve Redifer, and I happen to be the director of the HDIAC, and today we have the distinct privilege of hosting Dr. Christina Baxter for this presentation. I want to express my sincere appreciation to all of you for taking time out to join us today for this exciting and very relevant topic. And I'd like to thank our sponsor, the Defense Technical Information Center. If you weren't aware, the DTIC Information Analysis Center mission is to collect, synthesize, produce, and disseminate scientific and technical information. And if you haven't checked out DTIC's website, you can see it at www.discover.dtic.mil. I encourage you to take a look at their myriad offerings. And without their sponsorship, this webinar would not be possible. So a few admin notes as we begin. Please note that all the attendee lines have been muted. So if you have questions during the webinar, we're going to ask you to submit them using the text box that you're going to see on the left-hand side of the screen. And we're going to work to say the last 10 minutes or so of the presentation to go over those questions and discuss them with Dr. Baxter. Please note that this webinar is also being recorded, and a link for the recording as well as the slides will be available at the HDIAC website for later download for registered members. With that, it is my pleasure to introduce you to today's presenter, Dr. Christina Baxter. Dr. Baxter is a senior leader with over 25 years of experience in the chemical, biological, radiological, nuclear, and high-yield explosive defense community. She is well known in this community, and I gained familiarity with her work during my time as the commander of the Marine Corps Chem Bio Incident Response Force. Christina has served the public throughout her career. She was the manager of the CBRN program at the Department of Defense Combating Terrorism Technical Support Office. In that job, she was responsible for managing domestic and international CBRN research and development programs to combat terrorism, as well as overseeing international CBRN agreements with countries such as Australia, Canada, Israel, Singapore, and the UK. She currently utilizes her expertise to produce training courses, develop exercises, and provide solutions for emergency response through the development of next-generation tools that enhance situational awareness and first responder safety. Christina is currently the chairperson for the National Fire Protection Association Hazardous Materials, Protective Clothing, and Equipment Technical Committee, and she's also a member of the NFPA Fire Service and Occupational Safety Technical Committee and the NFPA Hazardous Materials Response Personnel Technical Committee. I highlight just a few among her many other selfless contributions to the first responder community. Dr. Baxter received her dual undergraduate degrees in chemistry and environmental science from the University of Massachusetts at Amherst, and she completed her doctoral studies in analytical chemistry at the Georgia Institute of Technology. It is our distinct honor to host her today, and with that, I'm going to turn it over to you, Dr. Baxter.
Thank you so much, Steve, for the intro, and thank you everybody for joining us today. I want to talk to you a little bit about biological threats. So we're going to start off. First thing, if you have questions, and we don't have time at the end for all of them, then please feel free to reach out either to Steve and the HDIAC staff or myself via my email listed on this screen, and it'll be listed again at the very end of the presentation. First of all, for those of you who have not participated in the HDIAC process in the past, it is really important that you participate and sign up for this, because I've worked with HDIAC for several years, written a couple of chapters for them and different things, but there's a whole lot of resources that they put together that is really critical to the emergency response community as a whole. I know it says defense in their name, but remember, we're all part of that same defense infrastructure, whether it be from the public safety or the military side, and whether it be domestic or international. So please join in working with our HDIAC friends and see where we can go from here. So I'm going to talk to you a bit about understanding and responding to biological threats. Now, when I'm talking about the biological threats as a whole, I'm talking about it from my perspective, from what I see in different chat rooms, different sources of information, mostly in the digital world. So as we go through, you'll see a few different screenshots and things of things that came mostly out of places like Telegram channels, other interesting sources like that. So I'm going to go forward and we'll start right in. So the first thing I want to bring to your attention is that the rest of the world, whether they be our friends or they not be friendly with us. They are watching what we do, how we do, and the things that are going on all around us at all times. So while we may choose not to watch our news stories, they watch them very carefully. And they actually share that information quite efficiently. One of the things that I want to bring to your attention is the topic of recent, where the title I would say is Now That the Facade's All Cracked. Okay, our response to the COVID pandemic, as well as recent domestic terrorism events, are being closely watched. One of the things that I continuously see is the highlight that now is the time to attack. Not because we have done anything wrong in our response efforts, but because we have so widely said that we are running out of PPE. We are doing different things that we don't have everything available to us. We're putting assets all in one place instead of having them spread throughout. All of those things are very watched by the people around us. And most of this communication, as I said, is digital in means. Back in around 2005, all of our information came from terrorist manuals and all those types of things. But that went away when the internet came around. And as social media became more and more prolific, the internet took over. It's very rare that we see a new terrorist manual that's not of a digital form in this day and age. We have to remember that the introduction of the internet brought fascinating and extreme benefits to society, including our increased connectivity, expanding our worldwide commerce, increasing our knowledge. All of those things that we wish we had when we were in school are now at our fingertips. But it also put in an area for very high levels of misuse. Starting in 2017, almost half of all crimes committed were committed online. And so when we start looking at that, we have to follow what people are doing, what they are saying, and the things that are going on around us virtually, as well as in the real world. Remember, it was only in September and October of 2020 that how to make a bomb was one of the top 10 Google searches out there. So that's the world we currently live in. Now we have to remember that in the international forums, while they focus on the United States, they also focus on our partners. So when I say our partners, the number one partners that they're focusing on are the US, Canada, the United Kingdom, France, Spain, Australia. Okay, obviously they also always target Israel as well. But all of these partners, what they're very much focusing in on is the public safety, military, and medical communities as easy targets. As the coronavirus spread throughout the world, there were all kinds of different, I call them posters, you can call them memes, you can call them whatever you want, but different pages and different graphics that were posted throughout. The one that you see over to the left, obviously a soldier of Allah, makes you think automatically that this is something that is very much a jihadi type of thing. But if you look over to the right, this was in Hind magazine out of India. So India even had their people targeting our US way of life. But it's not just from outside. If you look at the domestic forums, we have a radicalized group of people in the United States on either far left and on far right that are very much focused on ways to attack politicians, police, and the medical community. So where we start and we replace on the domestic front, we generally don't see as much anti-military as we do anti-politicians. The police and medical community are still there. But this was a meme that was taken from a regular CDC poster. And in this time, I'm sure the CDC did not say spend time in diverse neighborhoods and spend the day on public transport if you felt you had the coronavirus. This actually came from one of the white supremacy channels running on Telegram with a US base. And so as we start looking at that, we have to consider both our international and what their focuses are, as well as our domestic. And also remember that it's very rare that we see people talking about bringing a new threat into the US. It's about taking advantage of the threats that are available already within the US radicalizing the people that we want to do harm and have it go forth that way. So we need to focus always on what the emerging microbial threats are, because that's just an easy access point. So while we saw people talking about the SARS-CoV-2 or the coronavirus, as we saw in the last few slides, remember it wasn't that long ago that we were dealing with people in Spain, 4 people being arrested and put in jail for trying to figure out a way to get people to become human vectors of the Ebola virus and put them on planes. So all of these things, it's always kind of the threat du jour. And so I want to remind everybody that it's about preparing for the overarching threat, not necessarily focusing in on one in particular. Now, the threats have very much changed over the years. We now see a focus on emerging microbiologicals, so the pathogens that we can easily get our hands on and isolate and purify, as well as those things like the toxins that are easy to produce. I would say that most of our people are going to go for ease, and ease of making and ease of access, as well as something that is going to last around a little bit of time. But remember, as we also had the internet, we also at the same time had international travel become readily available to almost every citizen. We have people that now travel the world who a generation before wouldn't have been able to even go on a plane. So we're starting to look at this mass globalization, both digitally and with human movement, that is really affecting the way that we're doing things. So let's focus in today. From an operational perspective, I like to go to, it's called Global Incident Map. I use the free online version. I know you can buy more detailed versions, but all this is doing is a poll across the internet and across all media types saying, what are people talking about in terms of infectious disease outbreaks or global outbreaks of different types of materials? This is one of the first times I've done a biotalk in recent years where Ebola is not actually going on anywhere in the world. So that was actually very heartening to see, because it has been a problem for the last few years, and it was only late in 2020 when the last outbreak was contained. And not by contained, I mean there were no new cases within a 45-day period. So when we look around the world today, the things that are going on, anthrax, cholera, foot and mouth, Rift Valley, polio, dengue, monkeypox, I call HXNY, all of the different avian influenzas and the H1N1s and all of those, swine fever, hantavirus, Lassa fever. Those are going on in different places around the world. In the US and around the world, we have the coronavirus, obviously, but in the US, we also have equine encephalitis going on in Florida, which is where I live. So when we start looking at these things, the questions we have to remember to ask when we see a patient or we see a person are, do they have a fever and have they traveled recently? Those are the first indicators we have of something that could be infectious. So did they travel? Do they have a fever? Start there and start working your way out. But you have to understand what's going on around the world in order to be able to tie those things together. If we have to wait until a virus has spread a great ways, then we're really in an epidemiology approach where we're going to go backtracking. If we can't get ahead of that, we'll always be chasing that disease. So the current hot topics, the ones that I see or hear people talking about the most, still remain targeted assassination tools or small to mid-scale terrorism tools. And by that, I mean the toxins. So when you look at this picture, hopefully they jump to mind what they are. But as we start in the bottom left, we have Abrin. Going up towards the top, we have red tide or Saxotoxin. In the middle, we have botulinum toxin. In the top right, we have ricin. And then in the bottom right, we have Tetrodotoxin from pufferfish. So as we start looking at these things, I know all of you have heard of recently Abrin, ricin, and botulinum, but we have to remember that people are actually chatting about saxotoxins and tetrodotoxins more now than they have in years past. So we'll go into each of those, but one of the things I want to remember is that the internet chatter focusing on the use of biological weapons remains at an all-time high. This started going up in around 2018 and continues to this day. And the impetus behind most of it is if you say the same thing enough times, regardless of whether it's true or not, people would start to believe it. This is how radicalization occurs, okay? And so in this case, poster after poster saying that the United States used biological weapons in the Middle East, and now it's time to come back at us. So if you look at the titles of these, we will fight you with the same weapon you use to kill innocents. You will see the danger of your weapons in your own country, and the silent terror has just begun. These were posted around and continuously are posted based upon all of the different types of radicalization into it's okay to use a biological threat because they used it against us first, which we all know not true, okay? So as we go forward, they started following this up. So in July, we started seeing all of these posters, but as we started going forward into August, we end up with a whole series of how-to videos coming out, releasing very detailed step-by-step processes like a YouTube video on how to isolate, purify, and employ any of these types of animal and plant poisons. And so going from where we used to see things years ago in a written media to now a video step-by-step detailed process of how do you do this. And it gets away from us now that before we always assumed you would need a PhD microbiologist to take care of some of these effects. Not so anymore. So they go through the whole process from acquiring specimens. We used to always hear about soil from sites of recent outbreaks. Think of things like anthrax, not easy to isolate from soil. But reality is focusing more on commercial cultures, stealing it from universities and hospitals. There's a whole slew of different ways to acquire specimens, and they focus in on that. Providing the technical information. So whether it be the internet or technical journals, the Journal of Visualized Experimentation came out in 2012, and that really gave the people out there the information on how could you step-by-step do these, what we always considered very detailed microbiological processes, but no longer, step-by-step. We can access the equipment we need just by the DIY biology programs, the do-it-yourself stuff. So most of the stuff that we need, same type of information, the same type of materials that we're using to make beer at home, make wine at home. So you have all of this type of material readily available and no longer hard to find, okay? And besides, you can steal it, but we also have just buying it straight from any different places. The ability to culture and propagate the materials, okay? We always felt that you'd have to have that expertise in bacteria and viruses to do that. But in this case, today, they step-by-step tell you what's the best type of material to grow it on, how do you isolate it, and then multiple different ways in which you can weaponize it. For instance, there's one talking about botulinum that goes through a detailed approach on how to aerosolize it. And at the very end, they say, and if that doesn't work for you, go ahead and put it in solution and put it across the salad bar. So you start looking at these types of things and realizing that where we were on, hey, we want to spread anthrax around, now we're at the point where we're saying all of these things are things that you can do at home or in your own location, and you can propagate, weaponize, and disperse the threat materials. So I'm going to go through each of the different threats right quick and not go into too much detail, but I put these charts together just to kind of give you a sense of how each of these compares to one another in terms of the toxicity. So first off, we'll talk about ricin. Of the 5 we're going to talk about, likely the least toxic. Likely also the most likely, because ricin, I think about 95% of the world's ricin calls happen here in the United States. I don't know if it's because it's ease of access, because of the simplicity of it, but we actually like to have ricin calls here in the US. So the poor folks from the FBI are running ragged trying to deal with this one. Now, one of the issues we have is people don't understand the ways in which you have to get the toxic dose. So for instance, most people that deal with ricin are talking about it from an oral perspective. If you were to look at the LD50 or the lethal dose for 50% of the population, that's 20 milligrams per kilogram, and then look at that from the perspective of your average human, they would have to eat 1.6 grams of ricin and then die over 85 hours time period. I surely don't want that to occur, and the way that they die is not a simple, easy death. Now, for those of you who aren't used to the science side of this and getting into the grams and the milligrams and the micrograms, think of it from this perspective. When I have 1.6 grams, that's a bit more than a sugar packet. So when you go to put a packet of sugar in your coffee or tea, that's a little bit over a gram. Now let's let our hands be a little bit sweaty because, you know, I've got the heat on down here in Florida today. If I poured that sugar packet in my hand and then flipped my hand over and let the sugar fall off, I would have this thin film on my hand. That would estimate be around a milligram, okay? A thin film of sugar. Now, let's pretend it's salt now, just for perspective. Now we're gonna look at that and say, if I just took my two hands and wiped them across each other 3 times, not using any water, not using any paper towels, nothing absorbent, that's going to leave me with a few crystals on my hand. If the light hits those crystals, I will see a little bit of a glimmer of that crystal there. The minute amount that we can see with the naked eye is a microgram. Okay, that's what we're talking about here. And if we start talking about nanograms, that's after we've taken a paper towel, wiped our hands off really good. And the reason I told you to stick to salt now is because if I licked my hand, I can taste the nanogram of salt. I wouldn't be able to taste it if it's sugar. So putting this in perspective, 1.6 grams is a pile of material. If I inhaled it, it's 240 micrograms. That's still a lot of material. And if I injected it, 1.9 milligrams, a lot. So when we start looking at that, ricin as a root is really not the one that people would be using as much. But if we look forward, I like to use this one from Germany because it kind of looks at a different approach. And in this case, there was a plot going out, and there was 2 people that met on Match.com for jihadis. So they were both radicalized already when they met on their online forum and later got married. But what they were trying to do was produce devices like a pressure cooker bomb, but putting ricin inside of it. And so in essence, they were planning to have a 2-phase effect. First phase, they have the device and the injuries caused by the overpressure and the fragmentation of the device. But then by adding ricin to it, they were hoping to get ricin onto those particles, that fragmentation, and get that to be an injection type of threat. In terms of an inhalation threat, they really wouldn't have done much at all. In terms of an injection threat, that might have been more likely. I think looking at all of the data on the materials they had quite a bit. The bombs that they were making and the way that they were doing it, their likelihood of success in measuring success for them as in total kills was very low. But still, the fact that people are talking about it and attacks are being planned and partially implemented is the problem. Now, let's go forward to just this month. Okay, so in January 15th, there was a Creighton dorm in Nebraska where a student was making ricin to kill herself. Now, in this case, as we just said, killing yourself via ricin is going to be a long, painful death, and it's going to take quite a bit of material. Obviously, once she started this and attempted to harm herself, she called for help and ended up going to the hospital. But now we have a dorm, a college dormitory, full of students with ricin in it. And now how do we deal with that? And these are the types of things we keep seeing time and time again. There's still, I don't think we'll ever be beat up by the one in Vermont with the people in the adult living place where one of the ladies was making ricin there. And it's like, Don't be surprised. You can find it anywhere. I would say we find it mostly in hotels and in apartments and other things, but hey, we see it in dorm rooms. We've seen it in Vegas. We've seen it in over 65 housing areas. Okay, when we go to abrin. Abrin is derived from the rosary pea. The rosary pea is a little bit harder than when we're dealing with the castor bean, but in this case, it's quite a bit more toxic. So when you look here at 0.8 milligrams, and we were at double that for what we needed for ricin, but we're still at pretty high levels when we start looking at it. Again, no dermal threat at all, but it will be an inhalation, injection, and ingestion threat, taking quite a bit of material. The one that I like to focus in a little bit more on is Saxotoxin. So if you're not familiar with Saxotoxin, this is the paralytic shellfish poison, most commonly associated with shellfish and the red tides. So I had worked with this one a lot back in the mid to early 90s, working with National Marine Fisheries, trying to figure out ways that we could test for this besides having to capture a bunch of clams and put them into a blender and try to extract the material. Fast forward to today, the methods are published on how to produce this without having to have a shellfish. You could actually just make the chemical because it's a toxin, it's naturally occurring, but you can actually synthetically produce it as well. When we get down to the amounts of material that are toxic here, even when we look at the oral dose, we're talking about 0.5 milligram or 500 micrograms. Injection and inhalation, we estimate at around 50 micrograms. So very low levels. Now, lethality on this one is not as lethal in terms of it's a paralytic toxin. So it's more of an incapacitating type of agent. However, we have to remember that there is also the issue with paralysis of your diaphragm and paralyzing your ability to continue breathing. So really want to remember that this is one that is out there and that is being talked about quite often. Tetrodotoxin, or the one that we generally constitute and think of with pufferfish, this one is out there as well and is also in the micrograms per kilogram range in terms of dose. You start seeing symptomology about 30 minutes and it can be delayed up to hours. Very rarely is it fatal, but symptoms are usually present and you're starting to get that paralytic type of process as well. This is one of the ones we worry about because you can have up to 500 milligrams at the universities without any type of regulation. So something to keep in mind and something to know if it's in your area. Oops, sorry. Botulinum, again, botulinum is the oldest that we've talked about, and it's also the one that is by far the most toxic and the one that we should be worrying about the most. I would fully suspect that in the next year, we'll see an increase in botulinum cases around the world, not because of any type of overt attack, but because so many people tried their hands at canning while they were home during the coronavirus, that I fear that we will have some mistakes in canning, which generally leads to botulinum. There are natural outbreaks of botulinum every year in the United States, a lot of outbreaks in indigenous populations in Alaska. And so we're expecting those always, but keep in mind that it can be an approach where they are trying to spread it, or it could be something that is just naturally occurring. In this case, we have to remember that the inhalation, oral and the intramuscular or IV are all in that nanogram range per kilogram. So we are an order of magnitude lower than the next closest. The symptoms are actually relatively slow and occur over a couple of days. We often can look at the eyes and get the first indicators of botulinum toxin. You want to be looking for this and being able to identify it as early as possible, because many of the implications are long-term if they happen to you. So as the symptomology starts to progress in the body, there's a lot of things that we can't reverse. Now we have to remember that Shrinkio, so remember back to the Tokyo attacks on the subway, they also had botulinum approach in 1990. So this is not something that is new to us. It is something that we've had to deal with for years. But one thing I want to remind you is that it wasn't only in 2018 that we started hearing about something called pataluminum. And there were all kinds of posters around. We were trying to figure out what in the God's name is pataluminum. It wasn't something we had heard of. And the posters were talking about will make you fear the air you breathe. They were very focused on bioterrorism, but we couldn't figure out what this new agent was until they followed up, like they do, with new methodologies. And these methodologies showed that pataluminum in all the cases was actually botulinum. And it was a result of phonetic translation that caused the confusion as to pataluminum versus botulinum. And it was method after method of how do you produce it, how do you isolate it, how do you purify it, how do you then make it into something that is suitable for dispersion, and then how do we get it dispersed. Now, I wanted to just put this one up here to remind you that even though we're talking about 5 different toxins, we have to remember that botulinum is by far the one that is the strongest toxin that we have to deal with. So we've always focused on botulinum, just like we did with anthrax and a couple of the other very focused in ones. So nothing new here, except for that we have to keep our eyes open for a few more threats and a few more types of things that we should expect in terms of symptomology across all of these. Just remember, botulinum is going to be the strongest, followed by Ebola and Saxotoxin right there together, followed by Tetrodotoxin, followed by ricin. Okay, so strongest to least strength as we go through. Now I want to talk the rest of the time about some of the advances in biotechnology, because this is where we are seeing a lot of threats coming in. And the issue that we have here is that synthetic biology in its own is fantastic breakthroughs in science. But the problem we have is if we're not paying attention to these breakthroughs, we could very well find ourselves in some severe problems. Thankfully, we have a few different federal level programs that look very closely at this and have thankfully caught a couple of potential problems before they became large-scale problems. But synthetic biology has really blurred the lines between our traditional weapons and the weapons of the future. And this is where we need to be focusing a lot of our time. So those knowledge barriers that we used to have are gone today. So if we just look at the National Academy of Sciences Working Group, when they went through and said, what are our concerns? And what are our highest concerns down to our lowest concerns? So recreating or synthesizing known pathogens, the viruses, that is one of the top concerns we have. Making the existing bacteria more dangerous. So how can we genetically modify it to make it actually be more problematic? We saw this in 2012, which is what brought a lot of this to the forefront. And in this case, there were 2 different universities doing research under some NIH grants, so National Institute of Health. And when they did this, they were trying to publish some of the results, which brought up the dual use concerns. And because in this, they were looking at avian H5N1 and a genetic modification, which allowed them to increase its airborne transmissibility. As you can imagine, anything that increases the airborne transmissibility of a pathogen is something we do not want to see published. And this created a really intense discussions at the federal level on where do we draw the line between censorship and science. And I remember being in discussions on these 2 papers and trying to figure out if they are released, are they going to create a danger to the United States and our security? And the answer was absolutely. If these are released, they could very well increase the threat to the United States. And so changes in the way that we do grants and things that you're allowed to publish and not allowed to publish had to be put in place because of this very thing, which was dual use concerns by increasing the transmissibility of a disease through a genetic modification. Very serious. That brings us to this second level of concerns. We're making existing viruses more dangerous. Again, we're talking about all the different ways that we can make things worse for us. Now, also talking about ways that we can exploit the natural metabolic pathways. So manufacturing chemicals or biochemicals that are naturally found. Manufacturing chemicals using novel metabolic pathways. We get into the one that I think all of us hear and think, my God, that's long-range stuff, but it isn't anymore. But modifying the human microbiome and modifying the human immune system, modifying the human genome. We have talked about all of these things in terms of cloning and other stuff, but it's always been that pie-in-the-sky research of very academic, not operationally relevant. But when you look down at it, it is critical that we start following this, because remember, in 2017, we said earlier, over 50% of crimes were committed digitally. As we start having more and more access to this type of information, as well as the DIY bio types of approaches, we now have ourselves in a point where we could be having bio crimes and the future of crime becoming something that could overtake us pretty quickly. Now, we have to remember as well, we no longer need to have animals to cultivate and to make sure we get these materials to replicate properly. We have human organs on chips now. Okay, this is something that has been worked on and is amazing and very much needed for the scientific community, but also needs to be regulated in such a way that it's not out there and open for anybody. So we have small amounts of material that emulate that organ level function, and we've got to be careful about what we do with it. Okay, so when you're responding to any type of laboratory, keep your mind open that it could be anything. We talk about polylabs all the time today in our CLAN lab types of courses. And what we mean by polylabs is if they're producing one thing, the likelihood of them producing others is high. So when you have people who are enthusiasts at making explosives, well, if they can synthesize the explosives, they can synthesize other things. In many of the cases that we've seen in the last few years, where you're synthesizing drugs, you're synthesizing many types of drugs, not just one drug. You very rarely go and find a lab that is just focused on the synthesis of one thing. Why bother when you have the equipment to produce many? So we have to remember when we go into some of these things that it could very well just be somebody's advanced biotechnology, do it yourself at home type of good thing. This guy here was making, trying to make human insulin as part of an open insulin project. But understanding what you're seeing in the types of components you should expect is critical. Now, this is getting towards the end of it here, and I know this is a very busy slide, but this is a paper that came out of the University College of London in October of 2020. And what they did is they went through and did a review across many different platforms of all of the research out there. And what they were looking at was, what are the different types of biocrimes that could be committed based upon the information they have today? Biodiscrimination, cyber biocrime, biomalware, biohacking, neurohacking, do-it-yourself drugs, genetic blackmail, and illegal gene editing were the top things that they came across. Now, when we focus in on these, we have to remember that all of these things are becoming easier and easier to occur. I don't know how many of you have put in your DNA to the 24andMe type of things, but there's a whole slew of different ways for the genetic profiles and getting different information. But how much information is too much information? how much information do you want to ensure isn't shared with others, and making sure that your specific genome is not shared. I mean, we have to be really careful about some of these things. And so I recommend highly that you go and look into this paper and the negative impacts of synthetic biology. Please don't think that I'm against synthetic biology. I 100% think it is very cool. It's the wave of the future, but we need to get ahead of it, and we need to be understanding what the negative impacts are as well, to ensure that we safely employ these types of methods. Okay, and it wasn't too long ago that the US military started looking into artificial intelligence in the Ginkgo Bioworks programs to start saying, how are we going to use some of this to get ahead of what could be coming to us? We need to remember that all of these kits and things have become lower and lower and lower cost, which means that it's open to more and more of society. That then tells us that we should be seeing bioterrorism on a rise, and we need to be able to defend against it. So as we talk about that, we need to remember that there are a lot of different things out there that, as somebody who's following the bio threat, we need to be focusing in on. One of those that I really like is a consensus report that was done by the National Academy of Sciences. I really like to go to a lot of the National Academy reports because they are done by a group of scientists who have pretty much, I call it, they have no dog in the fight. They really, they just are there for the academic interest. And this report is Biodefense in the Age of Synthetic Biology. And I think it's really worthwhile for everybody to start looking at that type of information. You can also look at the Apollo program for biodefense that just came out in January 2021 from the Bipartisan Commission on Biodefense, the same group that put out the National Blueprint for Biodefense 5 years ago and hasn't really been overly addressed. So this is a federal bipartisan group, and so I want to make sure everybody's aware of these and this new one that came out of where should we be going to respond to this threat. And with that, that was all I wanted to talk to you guys about today. In terms of the threat that we're seeing, remember that open source jihad, which is coined by the jihadi community, is all about bringing the threat in the area you have it. So you need to bring the thought process to the area where the material is already there. So it's no longer about trying to gather materials and bring them into our countries of interest. It's about using the materials that are already there, but radicalizing the people and having them employ them. So instead of having to go forward and teach people how to get stuff through safely, now it's about teaching them just how to employ it. The other issue that we had recently, which I thought was really telling of the times, Hezbollah, if you're not familiar with Hezbollah, is an Iranian-backed terrorist group that is focused, a lot of their folks are focused in southern Lebanon. They actually had recruitment out saying, hey, Americans, if you need us to teach you how to do some of these things, please feel free to contact us. When we have Hezbollah reaching out to the average American saying, we can help you with some of these attacks, we have a problem on the horizon, and we need to be watching for that. So Steve, I am going to turn it back over to you for any questions. This is my contact information. If anybody wants to get a hold of me, you can get me at Christina@hazard3.com. And this is my cell phone, 404-408-8779. Please text me first before you call, because usually I'm doing stuff like this. So thank you very much.
Thank you very much, Christina. I cannot tell you how many compliments you got on the presentation and the attendee chat to the presenters. It just was overwhelming, the response, and that is directly attributable to you, and I'm very grateful for that. And I'll start off and answer one that we were asked repeatedly. Dr. Baxter has been kind enough to share her personal contact information with you right there on the slide. And as well, you'll see the contact information for the HDIAC pop up in the left. Dr. Baxter does a phenomenal job and a lot of work all across the country, very well known in the community for doing training, and she's just very well respected. And I know she would be happy to respond to you and happy to put something together for you. Likewise, the HDIAC can also customize training packages for you. We work with the Guardian Centers of Georgia. I encourage you to check out some of the training products that we have as well. We're happy to work with you. For those of you in government service, there's a ready contract vehicle to get to the HDIAC, and also a ready contract vehicle that you could use if you wanted to procure Dr. Baxter's service through the HDIAC. So by all means, please do that. So without further ado, yeah, please go ahead, Dr. Baxter.
Bring up, this is just the intro section to a course that we teach down at the Guardian Centers. So we actually go through and teach and bring in some of the different materials and teach you how to destroy, how to detect, and all of that. So this is how I came to be with you guys today, was the course that we do at the Guardian Centers, predominantly a lot with the Marine Corps, but really about making sure people know how to identify the threat, how to deal with it, but more importantly, how to destroy it.
Excellent. Yeah. For those of you who don't know, Guardian Centers is a member of the HDIAC team. And again, they're always happy to provide. If you go on the website, there's a HME training video series that was done with the Guardian Centers and part of the HDIAC. And Chief Warrant Officer retired Kerry O'Connell has given a couple podcasts and a couple webinars as well. And radiation detection, I think, was the last one he did. So by all means. So without further ado, we've got about a dozen questions. We'll get through as many as we can. I promise you, though, we're not going to take you over past 1,300. So please stay with us, and let's see what Dr. Baxter's thoughts are on some of these questions. So the first one that was asked when you were talking about the threat and you were watching the threat chatter and the threat streams, have you gotten a feel that there are any specific targets out there that might be, obviously crowded areas or something we all have to be concerned about, subways, that sort of thing. But are there any specific areas that you think are of particular interest to the bad guys?
Well, there was a series of posters. Now, they didn't say that they were specific to bio, though. They were targeting, in the United States, the specific areas that the posters that they literally said on them were San Francisco, which they consider to be our technology hub, New York City, which they consider to be our financial hub. The other one that was up a lot at the time, which I thankfully have seen diminish some, was Virginia Beach. And that was, in which I know sounds crazy, but Virginia Beach became a hot topic right after the targeted attack using one of our teams over for an Iranian person. So because the news media gave out information about where people were located, it provided information that was problematic. And so, yes, we try to focus in on those large cities, but it's really not about that as much. One thing that we have seen is it's any large gathering of crowds. I didn't put in the ones that they had up on this talk for Christmas. Holidays are always right targets. This year, not as much because we weren't going out anywhere. But generally, any time that we have a Christian holiday, or any time that we have a large gathering of people, or if you're in one of our major cities. Did I lose you, Steve?
Yes, indeed. Okay. I've got you here. I just want to make sure you finish.
Yeah,
so
sorry.
Lawrence Roberge asked a question, a follow-up to your discussion about the ease of making and the ease of access. And he wondered if you had any other thoughts on that. You talked about ricin and the other ribosome inactivating proteins. And he specifically asked you here if you had any thoughts on Pochelin's potential as a bioweapon.
I could not quite hear what you said.
Okay.
On the last bit. There you go.
Yes. Okay, so he said he's interested in any other thoughts you might have on the ease of production, ease of access, particularly on the ribosome inactivating proteins. And he asked about if you have any thoughts on Procellin's potential as a bioweapon.
I have not focused in on them, particularly because I haven't had any chatter on that specifically. Yes, there could be potential problems, but I only focused in on the problems where the chatter brought me. Shall we put it that way? I'm not going to present something that could be a problem if we're not seeing it as a problem.
Yeah, absolutely. And so how about this? Another one of our listeners kind of asked about the level of expertise. So while we talk, sometimes a professional chemist such as yourself, we talk about the ease of access, the ease of making these things. What level of expertise is really required for something like this?
Well, if you're talking about the synthetic bio stuff, I would say high school education or more. If you're talking about just the toxins that we talked about, including things like ricin, abrin, and specifically botulinum, I would say you don't even need a high school education. If we're talking about Saxotoxin and Tetrodotoxin, I would think you might need the college education because the information is not as detailed there yet. And if you're not going to isolate it from pufferfish or from paralytic from the shellfish, then you have to synthesize it. So you're going to need that advanced college education for the synthesis part, unless you isolate it. Okay. But it's getting easier and easier, and it's getting to a point where the education that we used to see and need is no longer really needed for these lower end threats. Thanks.
So Timothy Rice asked an interesting question. You have a lot of experience, dealing with the NFPA and particularly the first responder community. You have a lot of experience in working with the Incident Command System and the National Incident Management Framework. Do you have any thoughts on how those can be applied to responding to a bioattack or perhaps a pandemic? For example, during the recent pandemic, would NIMS have provided, in your opinion, a suitable framework for response? Well,
the incident management system is the framework for response that we use and have used historically my whole career in the fire service. If you go back to the incident command system, it was widely used by California for the wildfires before it really became so common across the country and the world. But anytime you have an incident management system, whether it be the NIM system specifically or the old wildfire incident command system, it's about a span of control and about setting up categories of operation. Anytime you see an emergency management type approach to an event, this is exactly what occurs. So I would say that almost all of the entities that from the public health arena that employed a response actually did employ an incident management based response and then focused it in on the different categories. You may not see the same categories you would see in a fire response or in a chemical threat response, but as we start looking through from an emergency management to public health perspective, you would see a group that is responsible for the testing, a group responsible for the PPE, a group responsible for the, right now, for the vaccines. And each of these would have a span of control over the vaccine sites and then further down. I know my friend Michelle Chalsky in Florida has been doing a lot of this, and she's an emergency management specialist who just happens to really be good at command. And so, yeah, I think it's been used. You see it at the local level. You may not see it in that broader federal level because they're more providing the support to a local and state effort. Thanks.
Yes, ma'am. I think that's a good experience. And, my experience as a federal response force as in CBRN was exactly that, that local incident commander is the guy that's going to be supported. So I think your point is well made. So Arnold Pachman asked a question, is this mainly a local threat limited to the area of dispersal of the agent, or any of these threats national or pandemic scale? I think that jumps to mind is things like wheat rust and that sort of thing. What are your thoughts on that?
So thankfully, and I may be, I worry if we ever get to one of the agricultural ones, we're in trouble, because attacking our food supply is just not what we would need. However, as we look at most of the things they're chatting about today, they're focusing on those toxins. Those toxins are very much assassination tools or very small scale approaches to a broader spread. You don't see toxins replicate on their own. Okay, they're not something that is going to replicate outside of the person. So we aren't worried as much on that large scale event of something like a COVID. However, if you go back to 2014 and you look at those 4 people that were arrested in Spain and are still in prison because of trying to use human vectors for Ebola and spread it on planes across the world, that is the way things happen. We saw it with COVID as an relatively innocuous threat. I mean, don't get me wrong, it's killed hundreds of thousands of people. However, if it was Ebola that had been on planes and spread this way, because we did have planes spreading COVID with people and people as the matrices, not the plane itself, but having a world environment where we travel, anything that's infectious can become that type of thing. Thankfully, people aren't talking about that outside of the what if.
Right. And I think in large part the literature that we read here is that the purpose of a terrorist attack, regardless of the weapon, is to do exactly that, inspire terror and mistrust on the part of the populace. So
that's
sometimes
right.
Those small localized attacks, yeah. I think that's exactly what our experience here and our research here is showing. So I think that's all the time that we have left for questions today. Again, thank you very much, Dr. Baxter. I can't tell you how much I appreciate you spending the time with us today. For those of you who don't know, Dr. Baxter and I were chatting before we began, and on one computer screen with one headset, she was conducting training, and simultaneously she was prepping for this webinar, y'all. So I want to say to everybody how grateful we are that we were able to capture your time and how generous that you have been with that time. So thank you very much.
And I just want to thank HDIAC because, I've been a big supporter of the HDIAC program for many years. And I want to make sure that the community that I represent from the emergency response side of the house, that they know that you're there and that there's information available that they can lean upon for more training for their people, especially in this time where we're not having conferences and things in person. The HDIAC resource is a fantastic place to get more information for your people's training. So sorry to throw you guys under the bus like that, but it's about getting the training to our people in the field. And so it's one more resource that they can all have.
Yeah, we love it. And likewise, we can reach out to people like Dr. Baxter and our subject matter expert network to get you answers to those hard questions. So thanks so much for joining us today. And as I mentioned at the beginning of today's webinar, as you can see on the left-hand side of the screen, the slides and the recording of Dr. Baxter's presentation will be made available for download at the HDIAC website, www.hdIAC.org. And if you're interested in learning more about the IACs or in getting involved as a subject matter expert or presenter or simply expanding your presence in the user community, please feel free to reach out directly using the contact information on that left-hand side of the screen in front of you. So in addition to monthly webinars like these, we also offer monthly podcasts, biweekly email digests, as well as a technical inquiry service with up to 4 free hours of technical consulting. And then again, we can also do customer-funded work. Many of you asked about that in any of the technical areas via our pre-competed contract vehicle. So again, that contact information on the left-hand side in that chat box. And if you enjoy the products, please like us and follow us on social media as well. And then lastly, be sure to join us for a really interesting topic that's coming up on 18 February. We're going to have a webinar titled Introduction to U.S. Nuclear Testing. And that's going to be given by Mr. Alan Carr, who's the senior historian for the Los Alamos National Laboratory. So that should be fascinating. Again, thank you all for your time today. Thank you, Dr. Baxter. Best regards, and stay well all.